Okay, I call this, excuse me while I finish my cough drop. I'm calling this Echoes of the Jurassic because the Jurassic is frequently known as the age of dinosaurs, clearly from the popular movies. You know, that's really been popularized, Jurassic, dinosaurs and such. And I'm saying Echoes of the Jurassic because I'm saying that the dinosaurs are still speaking to us. They're still talking to us. Are we really listening or not? All right, let's start out with a little bone anatomy. Maybe Joseph should be up here teaching this, but in a little bone anatomy, collagen is a protein. It's the most abundant protein in your body, and it's by far the most abundant protein in bone. The reason for that is because collagen forms this nice little mesh-like matrix. And on that, when you're making bone, your body has cells that will drop calcium and phosphorus onto this matrix of collagen, and they'll make a bone cartilage matrix. And that's what your bone's going to look like. And there's going to be, depending on the location of the bone and the type of bone, it's going to have different levels of density. So that's just kind of a real basic 101 of bone biology. But we have to keep that in mind as we go through this. Now also then, because bone is composed of not just mineral, it's not a rock, it is tissue, we have to feed it blood. And so we got blood veins, then blood vessels moving through bone as well. Okay, when an animal dies, if fossilization is going to occur, what's going to happen is that at least the best fossilization occurs by groundwater moving into the bone and bringing with it things like silica. And then the groundwater washes out the calcium and the phosphorus. So what we have is mineral replacement. We bring out the calcium and the phosphorus and replace it with other minerals such as silica that are in the ground, that are going to be part of the groundwater. And over time then, when this fully mineralized, then we call this a fossil. And it's a replica of the original bone. Is going to have approximately the same size and shape, but it's not living tissue anymore. It's just going to be, in essence, a rock. It's going to be just mineral. This then is why it was very surprising that in 2005, a scientist named Mary Schweitzer published a paper in the journal Science where she had isolated from the femur of a Tyrannosaurus rex tissue. Now, supposedly, see, over a process of lot of time, you should have had total replacement of all the material in the original bone with minerals. So it should just now be a fully mineralized fossil. But it wasn't done fossilizing. It still had tissue. In fact, this tissue was stretchy. You could pull it, you could stretch it. Now, needless to say, this drew a pretty strong reaction in the scientific community. Like, whoa, what's going on here? What is this stuff? She followed it up very quickly with 2 other publications in Science in 2007, further demonstrating the presence of tissue, the presence of non-fossilized material. But one thing I find interesting is she was actually not the first. The discovery of non-fossilized material in dinosaur bones actually dates back over 40 years. For various reasons that I don't know, these were rather ignored or considered not important or what have you. Part of it may have been because technology was assumed to not be sufficient, that maybe we could just chalk it up to an anomaly. Maybe we could just say, well, you're mistaken about what you found. And so we have a number of publications in the last 40 years, and this isn't all of them. This is just a small sampling of them. But none of them really ever made any impact until Dr. Schweitzer's paper. Now, one thing about her paper, color pictures. See, up until this point, colored pictures were very rare in technical journals because they're expensive. Colored pictures, even with scientists, have a draw. Like, wow, I can really see that. I can really sense that. So I wonder if that was at least part of the reason why her paper drew so much more attention. Now, I was part of a team that went into the Glendive area in Montana, which is a very common area for dinosaur fossil hunters. And this, again, this strata is identified as Cretaceous, not Jurassic, but Cretaceous. This is called the Hell Creek Formation. And we dug for fossils. And what we found was a brow horn of a Triceratops. And this is the picture of it. Now, I'll explain it a little bit more as we go along. But the interesting thing with this brow horn is that's all there was. The rest of the dinosaur was not there. It was just this brow horn. Okay, and as I note, it was encased in sandstone. But it was almost 4 feet long and 10 inches, I didn't put that, but almost 10 inches wide. So it was massive. This Triceratops would have been the grandpa Triceratops. He would have been huge. What was distressing to our guides was we were not digging this horn up as a museum display. This would have been one of the biggest Triceratops horns in any museum anywhere. We busted it up. And it was just like, it hurt them to think about that. But we were interested in something totally different than a display. What we wanted to do was to look and see what's in this horn besides mineral. Now again, going back to our horn anatomy, one of the things we also see is that, remember I told you that you're laying down this mineral on this collagen matrix. A group of cells do that. And when these cells do that and they get trapped in the matrix that they themselves lay down, they become called osteocytes. Now they don't die, they're still living, they're still helping in bone maintenance, they're just trapped in the bone. And we see here an illustration where you see all the little black spots, those are representing osteocytes. And then the larger picture you see an osteocyte has a very interesting morphology to it. Here is tissue we actually extracted out of that bone, or out of that horn. This tissue, you could stretch it, you could pull it, it was pliable, and there's an osteocyte. Now, and there's some more tissue, more osteocytes. Then when you do electron microscopy, so you can look at it closer, we see very distinct morphological characteristics of the cells showing their osteocytes. And here's some that have been washed off individually. One of the things to point out on this is osteocytes have what are called filopodia that extend outward and then kind of dig in or attach into that collagen matrix. These osteocytes from the dinosaur brow horn are so pristinely intact they still retain their filopodia, but these filopodia have diameters less than 300 nanometers. Now, if you know microscopy, you know that is extraordinarily small. See, bacteria are far bigger than 300 nanometers. One bacterial cell is far bigger. That tells you how small these are, and yet here they are pristinely preserved. Just to give an illustration, here we have the dinosaur osteocyte, and here we have a typical osteocyte from a vertebra, be it a dog, be it a cow, be it a human. Very distinct morphology, and yet look at the dinosaur osteocyte. Same morphology, same characteristic, retaining same morphology so that it's able to be microscopically identified. Still looks like a modern osteocyte. Lest you think that we contaminated, say this is a human skin cell. They don't look anything like those osteocytes. See, so we're not inadvertently seeing skin cells. Right? There were additional studies besides ours. Mary Schweitzer and other groups have done additional studies where they've added to the evidence. They've found more pliable tissue. They have found then, again, these distinct cells, just as we found. They have found collagen fragments. Remember, bone has that collagen matrix. They found collagen. Still there. Remember, collagen is a protein. They're able to sequence the collagen It's still intact enough that they can do that. And in fact, they found other proteins. They found actin. They found tubulin. These are common proteins in cells, in vertebrae. Here, if you look closely, what you're seeing in the background, that's still the collagen matrix intact. And that becomes critical here. Remember that. We're going to talk about that a little bit more in a second. The collagen matrix is still intact. It hasn't collapsed. It hasn't decomposed. It's still intact from dinosaur. Now, there was a study that was reported on about a year ago where they took and extracted and found tissue and fragments of collagen from some fossils. And what makes this significant is these fossils that they used were not freshly dug, weren't spatially stored. They were fragments of dinosaur fossils that have been sitting on museum shelves for over 100 years. Still tissue, still collagen. They also think they might have found some intact red blood cells. And they think, in fact, that the red blood cell may even have, and that's what the arrow is pointing to, may even have nuclei still visible. Also, they feel that they have found intact fiber structure consistent with bone collagen matrix in dinosaur fossils sitting on museum shelves for over 100 years. Also, a study that came out last fall, to me, this is how you do science. This study is excellent. They extracted blood vessels, intact blood vessels from dinosaur fossils. Intact blood vessels, so you can't claim it's some type of contamination, you can't claim it's some type of microbe. Intact blood vessels, extracted collagen, tropomyosin, actin, histone, tubulin, myosin, all of those proteins that would be consistent with a vertebra blood vessel. Extracted all of them. But this is dinosaur. In fact, in one paper last year, they referred to it as the common preservation of soft tissue. This is not any big deal anymore. Yeah, it's in all the dinosaur fossils. Now, what this brings us to is the classic geological interpretation is that dinosaurs lived 65 to 120, 130 million years ago. And they lived in the periods known as the Triassic, the Jurassic, and the Cretaceous. I guess maybe 250 million years ago. That's the standard geologic timescale, the standard understanding of the history of this planet. So these dinosaur fossils they're working with are given dates of a minimum of 65 plus million years. In fact, some of them have dates of 80 or more million years. Still retaining tissue, still retaining proteins, still retaining pristinely preserved cells. Okay, Mary Schweitzer, who kind of started it all with her 2005 paper and has been very actively involved in it ever since, I think she gave a very good summary of where we stand. She said, quote, that leaves us with two alternatives for interpretation. Either the dinosaurs aren't as old as we think they are, or maybe we don't know exactly how things get preserved. Now, because the dating paradigm is so well established, and if you don't think so, go into any university science classroom tomorrow and say, I don't believe that dinosaurs lived 68 million years ago, and see what kind of response you get, okay? It's so well established that in Mary's alternatives, in all honesty, there's not an alternative. It's not comprehensible that dinosaurs aren't as old as we think they are. Therefore, there has to be some kind of preservation mechanism. So that's where the focus is. All right, so how do you preserve tissue for 65 plus million years? All right, well, pseudomorphing. Now, I mention this not because it's really offered as a mechanism, but because people seem to think it is. So I have to clarify. Pseudomorphing is a mechanism where when the animal dies, bacteria invade into the bone and they begin to eat the tissue, which is part of the decomposition process. In eating the tissue, they make what's called a biofilm. If you've heard of microbial or bacterial biofilms. If you've ever noticed the kind of the slimy film on the refrigerator leftover, you know, on the back of the refrigerator you forgot about, that's a biofilm. That stuff that kind of gets on your teeth sometimes, that makes your teeth feel real yucky, that's a biofilm. Okay, so these microbes invade into the bone following the death of the creature, and they start eating the tissue and making biofilm. The biofilm can form an impression of the tissue. So as the tissue is decomposing, the biofilm has an impression of it. By the pseudomorphing method then, so I show, for example, an osteocyte. By the pseudomorphing method, the biofilm would retain the impression of the osteocyte, but it would then itself become fossilized. So what you have is a fossilized impression of the tissue. The reason pseudomorphing is not how you preserve the tissue is because with pseudomorphing, it's not a tissue anymore, it's a rock. You don't have protein, you don't have viability, you don't have plasticity, and you don't retain that pristine morphological structure. Biofilms give you general impressions. But I have to say that because people get confused because they want to think that biofilms, or excuse me, biofilms, that pseudomorphing is a way of preserving tissue, and it's not. But what also points out is that pseudomorphing requires bacterial activity. Keep note on that for just a second. Requires bacteria to be there. Also, just because there may be limited oxygen, there are microbes called anaerobes. And remember in the last talk we talked about E. coli had a different metabolism when it was growing with and without oxygen. Anaerobes don't like oxygen. They grow without oxygen. And they're all over the place. I used to work for an energy company. There are microbes that go down thousands of feet. Say it's hot, there's a lot of pressure, and they love it. We try to convince the geologists that methane you're pulling out of those holes, it's not 50 million years old, it's 50 years old. See, that was counter to everything they'd ever been taught. Because there's microbes down there actively metabolizing right now as we speak. So we don't have in the ground a bacteria-free or microbe-free environment. It doesn't exist. And just because there's no oxygen doesn't mean that the microbes can't degrade it. In fact, little side, if you give me a moment, little side story. Anaerobic bacteria can degrade lignin. Now, lignin is a component of plant that is very, very chemically resistant to degradation. I mean, lignin, in the biological world, lignin is concrete. Very resistant to degradation. Coal appears to be a fossilized form of lignin. When in the late 70s and early 80s, the research was coming out that anaerobic microbes could degrade lignin, I heard a very noted microbiologist say, I cannot accept that. Do you know why? See, what I just say is a fossilized form of lignin. Coal? Yeah. If anaerobes can degrade lignin, there shouldn't be any coal after 250 million years. That's just the biology of it. Because even though lignin degradation is slow, 250 million years, they still would get the job done. In fact, you remember the spill of oil off the coast, despite the centrifuges bought from Kevin Costner, which was just a publicity stunt, it didn't do diddly as far as the ecology goes. Who's cleaning up that oil? Microbes. Absolutely slow, but they'll do it. That's what they do. That's what God designed them to do. He couldn't accept that there was anaerobic degradation of lignin because if there was, he understood that means there'd be no coal. No, it just means they haven't been going 250 million years. Science at work. But see, that's rejected because we know the Earth is old. But here you have, we know anaerobes degrade lignin. We know there's coal. So what's the obvious explanation? Hasn't been 250 million years. Duh. Okay, so the right conditions are often offered then. Here's how you preserve dinosaur fossils. You've got to have the right conditions. Oxygen-free. Because see, oxygen is known to accelerate not only microbial growth, because microbial degradation of lignin aerobically is very quick. Anaerobically is slow. So get rid of the oxygen so it slows things down. Also, oxygen is very reactive. Do you know why the atmosphere isn't 100% oxygen?
Burn up?
Yeah, ask the astronauts of Apollo 1. Tragic story. And they finally said, why are we doing pure oxygen in a space capsule? We don't need to do that. Because if it's 100% oxygen, you light your match and you destroy the whole world. Whew. So what is most of the air? Most of the air is nitrogen, which is inert. Can't ignite it. So oxygen is highly reactive. So get rid of oxygen. Okay, oxygen-free. Microbe-free, because microbes are going to eat it. So got to get rid of the microbes. Okay? Water-free, because water first will bring in the oxygen and the microbes, and also water itself will accelerate reactions. Because water accelerates, especially protein. Protein's really vulnerable to water. Okay? Buried in an ash layer, that's much better than being buried in porous rock. Because first off, that limits the amount of oxygen and water. And if you really can control it, you want to bury it in a freezer. You want to put it in permafrost. Because just like your freezer at home preserves stuff, cold does also. So these would be your considered your pristine conditions for preservation by some people. Okay. Well, first off, most all of the tissue-containing fossils were buried in sandstone or mudstone. Because that's what most of the crustaceous in the Hell Creek is sandstone or mudstone. As I mentioned before, our Triceratops horn was buried in sandstone. Sandstone and mudstone are very porous. They're not going to trap or keep out oxygen. They're not going to keep out water. They're not going to keep out bacteria. They're very porous. All right? Microbes, as I mentioned, they exist for thousands of feet down. There's no area where these fossils are that there aren't the potential of microbes. Okay? Infiltrating rainwater and percolating groundwater are going to be all around these fossils. And you also, if the fossil is close enough to the surface, you have the potential of temperature fluctuations. If anybody here has worked with proteins in a laboratory, the one thing you quickly recognize is you cannot expose them to fluctuating temperatures. You either keep them warm or you keep them cold. They'll decompose faster warm, obviously, than cold, but if you cool, heat, they'll decompose even faster. So when you have fossils exposed to temperature fluctuations, you're actually accelerating the rate that this tissue and the proteins in it are going to decompose. Right? Here's the area where we found the Triceratops horn. Notice it's sandstone, it's not a layer of ash. Very porous. Also notice it was only a foot down. Now it took us 3 hours to dig through to pound through sandstone, because that's solid rock. I mean, we chiseled for 3 hours on that. I'm glad it wasn't 2 feet. We may never have gotten it, but it's only a foot down. Very exposed to temperature fluctuations. And in that area in Montana, it's going to get pretty hot in the summer, pretty cold in the winter. So there you go. All of a sudden, the very antithesis of pristine preservation conditions. Also, when we pulled it out, it was wet. See, percolating groundwater, infiltrating rainwater. See, when you're just a foot down in that sandstone, rainwater is going to come down. In fact, plant roots were coming down into it as well. Right? So there was nothing even close to the desired preservation conditions for our Triceratops horn. As I mentioned, temperature fluctuations, and hardly microbe-free. There's no such thing as sterile mud that I'm aware of. And no such thing as sterile sandstone. Oh, and also one other thing, my pointer, the base of the horn was actually exposed right here. It was exposed to water. It was exposed to mud. And in fact, it was wet and muddy. And Mark Armitage, who was also on the team, I turned to him when we were pulling this out, and I said, there's no way we're going to find anything in here. And I wasn't even assuming it was millions of years old. And I was still saying, there's no way we're going to find anything in here. Because it was so ungood conditions. And yet we did. Now, I'm sorry. I don't care who you are. You are going to have to give me a lot more than you've given me so far for me to be able to believe that that horn in that location with that level of exposure somehow survived 65 million years and still had the tissue and the cells in it. I just can't do it. That defies everything I know about biology. And it defies everything you know about biology too. Just doesn't work. I don't care what your preservation model is. 6,500 years is a stretch. All right, another method that's been talked about is called collagen sheltering. The phosphate minerals in the fossil, which we call apatite, are going to be tightly packed around the collagen, and they'll keep it sheltered. What happens is when collagen expands, it becomes more vulnerable to microbial and enzymatic decay. So you want to prevent expansion. So this apatite surrounding and packing the collagen prevents expansion. Preventing expansion then minimizes the degradation. That's how the model works. And in some ways, that's true. I mean, there's nothing chemically invalid about this, except, of course, whether you really can get prolonged preservation. You can maybe get 6,500 years of preservation, but not 65 million years of preservation. Okay? See, so less expansion, less degradation. There's been a number of decay studies done where they've established rates of decay for collagen, because this involves not only archaeology, it involves forensics. How fast is the body decomposing? What's going on with the rates of collagen decomposition? So we can identify times of death. We can identify how long it's been there. So there's been all kinds of studies done. What I found interesting is up until 2005, no study said you could get anything even close to millions and millions of years for collagen. See, and after 2005, all of a sudden, oh, we need to refigure all this stuff. But they took into account the apatite effect. They already took into account that it had the sheltering when they did these models. Also, you cannot reproduce what I'm calling the extraordinary preservation. The most that any model has ever shown, and that's when you give it every chance to succeed. Give it the most pristine preservation conditions possible. The most they've shown is collagen would last about 1 million years. Now that's 65 times too small. Also with collagen, remember you don't want it to expand. One of the ways you keep it from expanding is you dehydrate it. And you would assume if in a fossil in a very dry place, it will dehydrate. But as you remove the water, it also becomes less stable. You start losing that matrix. Remember I showed you those wonderful pictures of the collagen matrices that are being found in dinosaur tissue. When you dehydrate it, you start destabilizing that. So what's that tell you? It tells you that the collagen in these dinosaur fossils has not totally destabilized, which I would interpret means that they're not 65 million years old. They maybe could survive a few thousand years without totally destabilizing and coming apart and losing the matrix, but 65 million years? I know no chemistry to support that. In fact, the chemistry says the opposite. The chemistry says it will, after 65 million years, destabilize. Okay? Said none of this, as far as excessive, prolonged preservation, has been ever demonstrated. Collagen, in fact, has a half-life at 7 degrees. Okay, those of you that are maybe not real familiar with centigrade, because in science we use centigrade, 7 degrees is equivalent primarily to what temperature you're more familiar with? Refrigeration, although that's kind of a warm refrigerator, but refrigeration temperature would be about a warm 7 degrees. Yeah. So in other words, if you put collagen in a warm refrigerator, you'll get a half-life of about 130,000 years, which means that after 130,000 years, you've lost half of it. Another 130,000 years, lost half more. And that's where they come up with no more than 1 million years. But remember, that's under those cold conditions. None of the tissue-bearing fossils are at 7 degrees constant. Yeah, they're going to be in the fluctuating, they're going to be in warmer temperatures most of the time. So collagen degradation is going to occur a lot faster. Okay, iron model. This is by far the most popular model. This, in fact, this is the model referenced in the Jurassic World movie last summer. You know, well, just use iron to preserve. In fact, a little aside again. Remember the original Jurassic Park movie back in 1993 that all my students were coming to me wondering when it's going to be we're going to clone dinosaurs? I said, well, you got to understand the movie took a lot of liberties. Okay, but where did they get the DNA? Do you remember?
Amber?
Yeah. Did you know they've actually shown that amber is not sufficient for preserving DNA? Which I thought was kind of interesting. Okay, if DNA is not adequately preserved in amber, how are you going to preserve DNA in something much less pristine of environment than amber? Okay, iron stabilization. The way what it's saying is that the hemoglobin in the red blood cells and the myoglobin in the muscle cells all contain iron. And when the creature dies, then these cells rupture and they release their hemoglobin and myoglobin, and that provides iron then into the tissue. The iron then will create a situation to preserve the tissue. And I'll explain how in just a moment. So they're saying the bone will have a high concentration of blood. The blood will have a high concentration of the myoglobin and hemoglobin, which means you're going to have a high concentration of iron, which means now you can coat the tissue in iron, and that's going to initiate this preservation process. This is the most popular model. All right, the way that works is iron initiates what's called Fintan Chemistry. Fintan Chemistry is where iron will react with the proteins and other molecules in the cell, and it will result in forming free radicals. If you had chemistry, you understand a free radical is not an ion. It's not looking for an opposite charge. A free radical doesn't care. A free radical just reacts with anything. And the hydroxy radical, for example, shown there, it is highly reactive, especially with proteins. And it just starts setting up reactions. That's why in your body, they tell you to drink antioxidants to control these free radicals, because they will do all kinds of reactions that aren't particularly healthy for you. But if you're dead, of course, they don't care. If you're dead, then supposedly the free radicals can actually preserve you. So they may kill you, but then they preserve you. Okay, so what will happen is the free radicals will form cross-linking in proteins. What that will do is, for example, the little circles represent amino acids in the proteins, because proteins are long chains of amino acids. The free radicals will set reactions to occur where the protein will essentially fold back on itself, and you'll then get a reaction where these two amino acids that are in the protein chain but not bound to each other now bind. And essentially the protein ties itself up into knots. And in tying itself up into knots, it now becomes less sensitive or becomes more resistant to microbial attack and enzymatic attack. And it is true. I mean, I'm not denying that this occurs. This is the preservation techniques you use when you do microscopy. You preserve your protein by creating these cross-linkings. So to demonstrate their model, they did a 2-year study where they took ostrich blood vessels and they soaked them. They soaked them in iron, they soaked them in hemoglobin, they soaked them in water, and then they looked and they said, okay, let's see how much deterioration has occurred over the period of the experiment. And what they concluded was that they had a 240-fold increase in tissue preservation or tissue stability of the hemoglobin-soaked ostrich blood vessels versus the water-soaked, demonstrating in their mind then that the iron from the hemoglobin is causing these reactions, thereby creating a more resistant protein so it won't degrade as fast, and so you have greater preservation. Here is some of the results. For example, blood vessels soaked in hemoglobin that oxygen-free, take the oxygen, because remember that was one of the criteria that some people felt was important, keep oxygen away, because we know oxygen will accelerate microbial growth and oxygen will accelerate chemical degradation. Okay? Versus blood vessels soaked in water, oxygen-free, and in their conclusion, we see the difference, nice and rich color and faded and pale. And then the same thing when they did in the presence of oxygen. Now, already one thing I would point out, you can't take color into account because when you soak blood vessels in water, what happens? They decolorize. So already what we see is a potential bias occurring simply because, it looks nice and red and rich and healthy instead of pale and kind of, weak looking. So you create a subjective, and this wasn't any kind of analytical evaluation. This was just simply a look through the microscope and make a determination. Very subjective. So what we see is we have the very distinct potential of a subjective evaluation based strictly just upon color differences, which have nothing to do with preservation. They have to do with your experimental condition. Now, these pictures were not published in the article, and I kind of found that curious. These were published in an online supplement to the article. These are electron micrograph pictures at 8,000 times magnification. This, again, is hemoglobin-soaked, oxygen-free, water-soaked, oxygen-free. They actually found out that in the absence of oxygen, they felt they got more deterioration, not less deterioration. So they've now concluded that you want oxygen for preservation, which is contradictory to what they had been saying. But remember, oxygen is known to increase microbial growth and known to increase chemical activity. So you got what I call diametrically opposed situations going on here. But what I found even more interesting was in the water-soaked and hemoglobin-soaked with oxygen, that they pronounced as hemoglobin-soaked with oxygen gives you the best preservation. I don't see any difference. In fact, let's blow it up. Here's your hemoglobin-soaked. Here's your water-soaked. I don't see any difference. Now, I'm not saying that's why they didn't publish this in the paper. I just found it curious, though, that this was on a supplement that most people weren't going to read. To me, this negates everything they were talking about at the lower magnifications, where they were really, I felt, just kind of taking into account the subjectivity of color. Now, they did some other analytical processes. Most of those were to establish the presence and absence and location of iron, not the deterioration of the tissue. And so one of the challenges I have is, why didn't you do much more elaborate analytical analysis of the tissue itself to determine what level of degradation you had going on, not just subjective visual? Well, that was, after years of very careful and meticulous study, all of a sudden it's like they just suddenly went back to some very one-on-one basics. Yeah, well, that's what, again, because they've been very good scientists. I mean, Mary Schweitzer, I would say, is a top-notch scientist, has done excellent work, but then it's like, okay, but I don't understand. I'm confused. Now, I understand they've been continuing the research, and we'll look forward to their next publication. But just based on this, just based on their own work on this, in the supplement, I don't see the difference. I'm not seeing anything. And I would assume, because you know when you do electron microscopy, you can kind of fish around and find where you want to take a picture of. I would assume they would have taken pictures of what they would consider to be the most degraded parts. Not, oh, here's one part that hasn't degraded, let's take a picture of that. You know, do you see the difference? I don't. I don't see the difference. But let's do some analytical. Okay? First off, 2 years at a constant temperature doesn't really give you too much insight into what happens for 65 million years in a non-constant temperature. Okay? Also, they admitted they got very little hemoglobin release from the red blood cells, so they had to use a blender, which of course all the fossils took their own blender with them, right? You know, because they had to rupture the red blood cells in order to get sufficient iron into the tissue. Most dinosaurs remains, most dinosaur fossils are what we call disarticulated. In fact, they're what I call highly disarticulated. Do you know what that means? Disarticulated.
Ripped apart?
Yeah. In fact, to me, they were pulverized. It wasn't they just died and a scavenger moved their bone. They were pulverized. That doesn't occur except for massive cataclysmic events. When you pulverize something, you're busting it all apart. What's happening to the blood? Yeah, it's going everywhere except into the bone. So they're not being buried in blood baths. Plus, we have tissue preserved from sponges, tissue preserved from beardworms that are identified as 500 million years. Think of that. 500 million years, and they're finding protein in these things. This is a sponge. This is a beardworm. They weren't buried in a bloodbath because they don't have blood. So how do you apply the iron model to that? See, it doesn't apply. Okay? Also, a paper that just came out, I mean literally just came out, they admit there's probably not enough iron in the dinosaur for the model to work. So they propose an external source of iron, at least as a supplement. But, to me, here's the real key. Regardless of whether the iron comes from inside or outside, if this model actually was the preservation mechanism, it would leave a chemical footprint. Because what did I just say is happening? How does it preserve? It preserves by tying the proteins into knots. It preserves by reacting with the amino acids in the proteins. Tyrosine and methionine are amino acids that are highly susceptible to Fintan chemistry reactions. So if you had Fintan chemistry occurring in these proteins to preserve them, these would certainly be target A for those reactions. And yet we identify tyrosine and methionine in the dinosaur proteins. They're still there. They're not altered. They haven't undergone any kind of reaction to alter their chemistry. They're still amino acids. That footprint is absent. Serine, asparagine, glycine is a highly reactive sequence of amino acids. Again, very susceptible to the Fintan reactions that are going to be occurring in the iron model. And yet, serine, asparagine, glycine sequences are found in dinosaur proteins. So what we have is a lack of the chemical footprint that should be there if the iron was acting the way that they are saying it acted to give you the preservation they're saying it should give. Now again, I'm not saying iron cannot give some preservation. I'm just saying that we don't have either in the presence of in the absence of a footprint or in the absence of blood baths, we don't have any reason to believe that it's going to give us 65 million years worth of preservation. It may give us a few thousand years. Plus, remember already the protein degradation models show that proteins are capable in some circumstances of surviving a few thousand years, but not millions of years. Jeffrey Botta, who is a geochemist, he rejects the soft tissue for a very good reason. He's an evolutionist. He's committed to the evolutionary time scale. The dinosaur fossils have to be 70, 80, 90 million years. They can't have tissue because bones absorb uranium and thorium like crazy. In fact, if you take a Geiger counter and go up to the dinosaur fossils and I've done it, you go up to the dinosaur fossils in Colorado and pass that Geiger counter over them, they'll light it up. They are radioactive because they'll absorb it like crazy. And so he says, because of that, you get any biomolecule wiped out. It couldn't be there. That's why he rejects it. Says it can't be real tissue. And there's not been any preservation model that's even talked about this. They just ignore it. Now, how's what I would call the more appropriate way of, or the more consistent way of accounting for this? It's not 65 million years worth of exposure. It's a few thousand years worth of exposure. So you still have some tissue remaining. By far the superior way of accounting for the radioactivity we know is there. So what we see is that there is original dinosaur tissue. It's pliable, it's stretchable. Okay, there is original dinosaur cells, in fact, in a pristinely preserved state. And there is original dinosaur proteins. There's many of them. We already named several. You can even do amino acid sequencing of them. There's lack of an adequate preservation model to explain how this could be preserved for millions of years. In fact, the protein decay models, the experimental evidence we have, says the opposite. You can't preserve it for millions of years. The tissue comes from fossils that were not preserved in pristine conditions. They weren't in cold, they weren't in oxygen-free or bacteria-free or water-free. They were in what we call very harsh environments. All of this is totally inconsistent with that these fossils are old, but completely consistent with that these fossils are young. And in fact, I'll let a couple chemists do the talking. They say that the survival of collagen and osteocyte peptide fragments is parsimoniously explained by a young age hypothesis. While counterintuitive to conventional thought, it's unavoidably visible. The primary way it's dealt with is just simply not considered. That's not an option. Remember, Dr. Schweitzer said, either the fossils aren't that old or we don't know how they're preserved. Well, they're not considering they're not that old. That's not even on the table. So we just got to figure out how it's preserved. But I say you don't have an adequate mechanism, therefore, why should I accept that there is a special preservation? When, if they're young, that makes everything fit. You don't have to do any kind of wild extrapolation. You don't have to do any kind of ignoring of the decay models. You don't have to do any kind of, well, there's some way that we don't know about. If they're young, then everything fits. So now we look at, okay, age of dinosaurs. Do we really have to go there? Or are we looking at evidence that says, no, we got to reconsider? And as far as radioactive mechanisms, radio decay mechanisms, again, I don't have the time, that's a whole nother talk, and I'm not a geologist, so it's better other people talk about this. But one thing I would point out, the basics of the geologic column and its time frame were established decades before radioactivity was even discovered, let alone the methods developed. So in the early 20th century, when they were developing the radiometric methods, you already had a geologic timescale similar to this. Did they use the radiometric methods to establish the timescale, or did they use the timescale to establish the radiometric methods? See, it's not surprising that they use the radiometric methods that they use, because they're not a collaboration of the timescale, they're a product of the timescale. And since I have just a moment, I want to show you one slide that I put in just in case I had time. One of the big things to talk about with radio decay methods is that it's so internally consistent. You know, it's cross-checking. They agree with each other, and they agree with all kinds of non-radiometric methods. And I get that all the time. Here's a paper that was published in 2009 in the Journal of Evolution, of all places. And their purpose of the paper, this is the purpose of the paper, was to, have you seen this paper? The purpose of the paper was to establish the nice continuity of all the dating methods. And yet, when you go through and you pick out their data, what you find is you get frequently a 90% and sometimes a 99% difference between different methods of the same location. And sometimes 99% difference of the same method in the same location. So which date do we pick? Is it 420 years or 114,000 years? Well, it just depends on what I want it to be. So, of course, then what do they use? Do you know? Somebody give me an answer. They use biostratigraphy. Biostratigraphy is fossil progression. And that's based on what? It's based on evolutionary assumption. So don't tell me that radiometric dating is somehow so objective and it gives you this nice analytical. It's nothing of the kind. It is calibrated by the fossils, and the fossils are interpreted by evolution. And for my last talk, remember I showed that there's no reason at all to say that they aren't totally misinterpreting the fossil record. So if you have a misinterpreted fossil record, then what's that do to your radiometric methods? See, it throws everything into extreme doubt. And so now all of a sudden, that's why I call it echoes of Jurassic. Now all of a sudden that tissue starts speaking to us and saying, hey, you can't ignore me. See, evolutionists have been ignoring it, saying, well, it's irrelevant if there's tissue or not because it doesn't determine the age of the fossil. Yes, it does. It very much tells you you are misrepresenting the age of this fossil, regardless of what method you're using, because that tissue is there for a reason. And that reason is either it's, as Schweitzer said, either that fossil's not as old as you think it is, or it's preserved by some bizarre method we don't know anything about. So we're back to that, where she correctly put us. But you got to bring onto the table the questionability of the age of the fossil. That has to come back onto the table. If it doesn't, then you're not going to get anywhere. And like I say, you can't hide behind radiometric methods because they simply aren't giving you what you're claiming they're giving you. Okay, with that, I will shut up. I told you it'd be a little shorter. It wasn't too much shorter. Well, now that particular samples, they weren't working with what they would consider to be millions of years. They're working with younger samples. But again, different methods give you different age ranges. And how do you know which method to use on your sample unless you have an estimate of the age to begin with? And where do you get the estimate? From biostratigraphy. So you'd say, well, I can't use carbon-14 on dinosaur bones, for example. They're too old. Well, I didn't know they're too old. Yeah, I have to already have predated them before I can date them. So what we see is even with these dating methods, we have to have an estimate of the age in order to know which method to use. Yes?
In some discussions, we talk about, we found carbon-14 in these.
I didn't go into that because obviously that would, right.